Yeah, I think it was just.
I don't know.
Yeah, it was.
So then I started screaming Central Dogma, and that's kind of when I remember everything going away. So what, pray tell, is that?
Is that the theory for transcription and translation? Wait.
That's the whole thing.
Oh, it's just transcription and translation.
Yeah.
Does it include replication?
No. Yeah, plans are changing in real time, so I'm trying to make this work for you.
So how's your demonic going?
I can never get
How much? How much work have you done?
Yeah.
Boy lost lacrosse, sad. He had a strong first period though, strong, good. Went to the Bayou City Art Festival on Saturday, really enjoy it, but Saturday was so sunny, I have a little sunburn, and I was in a race to get to set lacrosse game, so I didn't fully enjoy it, but it was cheap because I didn't buy anything, because I was in a hurry, so that was like, honestly a win. But it's one of my favorite days of the entire year is that festival. So that was my weekend. Sunday, I think, I don't know, not much happened. I watched a little TV, watched a movie. Good living. TV. I remember that. I'm on episode 6 of The Last of Us. There are 9, right? Like I'm catching up.
What are your thoughts? How do you like it?
I have enjoyed it. But it's kind of dark, right? So I don't like to watch it super late at night.
I don't like horror movies. Isn't it like a horror
Mind you,
thing?
I was in 3rd grade,
Sure.
so like,
If it has zombies in
it
it?
was terrifying, and like any creak in the door would be like, it was bad.
So I'm not worried about the mushroom people coming to campus, but like, there've been some really sad things, right?
It's very sad.
And that's what I don't want late at night, like
Like
it's,
in the game, it's like even worse because you get attached to the person, like you are that person.
Wait, what?
And then you're just like all like.
yeah, I don't do all this. All right, you played the game, I don't know that you watched the show, does anyone else watch it?
I watched clips.
I haven't found.
I know that Ellen's a
I've seen,
lesbian.
like, billions of it.
Well, what do you do?
Well, or maybe she's not.
Why is there HBO?
So we have Paramount because we've been watching some Picard. And then we have HBO to watch that, and then you got Disney because Mandalorian, and then it's like,
I pirate everything now.
Well-
It's like.
So I prefer it when Netflix was monopolizing everything.
I did too, it was so much cheaper.
And now Netflix has ads. Ugh. Yeah. Next, I think it's like in the next month, they're gonna take the standard account and make it $7.99 a month, and it has ads. So then they're forced to buy the next level of. And then they're also adding a new feature where you can't share passwords.
Yeah,
So if you're in like 2 separate locations that are
we do have a friend borrowing a Spotify family account, and I wonder when that's all gonna go to who.
Yeah, I've been.
Not my friend, I don't care. Okay, so what is this glorious thing? And how would I use it?
You would use this to do all the like codons for translation in order to make it
So if we were to find the problem that I gave you last time, I'm looking for my copy. You can feel free to look for yours or to assume that I'm gonna get it sorted in a second. You know, I have one for Elizabeth somewhere, I probably have one for Aditi somewhere. So here's a question that I think is phrased kind of poorly. Part of a strand of DNA contains the codons ACA, it's bad when you can't write the right letter, AAA, CTT, GT, GACT. Determine the amino acid sequence of this part of the protein that the sequence forms. What would I do? Okay, I heard I have to make it RNA, so I'm going to assume that this is the right strand. For those of you who might know that there is a complementary and there's, you know, this and that. Like, we're dumb, we're chemists. Let's just assume that this is it. What am I going to do to transcribe this into RNA? So I'm going to find the partner. So A is going to go to U because that's one of the distinctions between DNA and RNA. A, adenosine, pairs with uracil. So we could just write this out like we know what we're doing. Everyone okay with that? And then how would I use the codon chart that Brecken introduced me to turn this in turn into amino acids? So I'm just going to come over here, and this is the easiest flavor. The first letter is here, the second letter is here, and then the third letter is here. So UGU codes for cysteine. UUU codes for, yeah, what is it? I like it. Phenylalanine. That's what some people are, you know, allergic to. GAA codes for glutamic acid. CAC codes for histidine. And UGA does not code for puppy dogs. It codes for stop. So, you know, my non-biologists haven't done this in a few years. My biologists think this is trivial. I thought it was worth spending a minute reminding you how to do it. So again, we found the complementary base. So we learned last time that A and T pair, G and C pair, but a distinction between DNA and RNA was that T was replaced by U. That's just the loss of a methyl group. It has something to do with making RNA less stable than DNA. DNA persists for like thousands of years. RNA persists not that long, months. Did you hear the article about them finding Beethoven's hair? NPR had an article about Beethoven's hair. So they had some, and they found out it was hair from 3 different people. But then they found another sample of hair that was probably his, and they're trying to figure out if some of his health issues were genetic. So they're trying to figure out why he was deaf. And he also had some gastrointestinal issues, and they're trying to figure out if those were genetic. But like the persistence of DNA is such that you can do that. Now I watched Jurassic Park, you know, for joy, and DNA don't last that long. So that is ridiculous, but Beethoven we're still, you know, in the fold.
Do you know how deaf he was?
I do know, I believe he was profoundly deaf. Like, yes, crazy, right? Like, I know that people can read music and hear it, but I don't really know what that means.
Like even like the tiny, I don't know the specific word for it, but like nuances and intonation, like you have to like have like that good sense of pitch to like know if you're out of key and stuff. That's really important. No, wasn't Mozart like blind or he had like I don't know, there were a lot of composers with health
Like, he
issues.
got lots of, like, he was an alcoholic with a number of health issues, but I don't remember. Anyone can pipe in, anyone. Sorry, I don't know. Talk to, half the class about the joys that are BT corn. In BT corn, we take this juicy little gene segment from a particular bacterium that exists in a, what?
He wasn't blind, but he had like an eye infection and
Okay.
gave him temporary blindness. But Bach was blind, which is also crazy because he was a really good
So Joseph Saye is about like, what does it mean for a clarinet to play a note as opposed to a piano to play it? Like, what is 440, what does A mean, and how is it different for different instruments? And that's not really what it is, but that's how it started. It's a lovely YouTube video about that. So we clipped a gene and we inserted it into corn, and that gene kills these gross corn borer bugs, corn borer beetles. That look familiar? We don't talk about restriction enzymes. We don't talk about anything.
We did a homework assignment on this last week.
I taught this to first and seventh period. Like, I'm good at this.
Where we did
The learning guy, like he could talk,
Yeah.
like he could talk about BT corn is really good for genetic.
I did the homework. I don't
I mean,
remember.
you didn't have to. It was just like give an example of
Was it not
gene modification,
3.5?
and I just mentioned that.
Yeah. I would too, because you learn about it in 3.5 and in the options, so it's something that you like legitimately have to know. And then it comes up in your chemistry class, like this is the example for those of us who don't study this. Is this good or bad? So 2 benefits of GM foods are that they can enhance flavor, texture, and nutritional value, also increase the resistance to disease. 2 concerns are that not enough is known about how genes operate. I don't know about that. So there may be unforeseen consequences, and genetically modified genes may escape to contaminate normal crops. State 2 more benefits and 2 more concerns of genetically modified foods.
There's a tomato.
What? No, that was on the homework that I gave you last time, so I was reading the question, like the slide, whatever, the slide has the answer, so I was just waiting for you guys who, had a homework assignment about it last
We
week.
can remove like this stuff that people have allergies to.
But it can also put in something that you could potentially be allergic to, like corn all your life has been fine, and suddenly you can't have it. So good things, it could last longer, it could taste better, that was something you couldn't use, it can be pest resistant, it can increase the amount of vitamins go to your golden rice, it can increase crop yields. And it can be drought resistant, salt tolerant, whatever. Just have to put this in your brain somewhere. For many of you, it's already in your brain. Girl Emma has to choose 2 of these that she wants to remember. And then if we look at the other side of it, some things we might worry about. We don't know what the long-term effects are. We don't know how we're gonna change the nearby plants due to cross-pollination. We could be adding allergens. We're changing whatever makes food food. We could be making superbugs. And we do not know how to label food appropriately. Because it sounds scary, right? Why do we call it an MRI? Because nuclear sounds scary. We don't want to do a nuclear magnetic resonance image of your knee, but it's the same technology. How do we make this honest but not scary? Genetically modified food, is it going to genetically modify me? I don't know. I don't believe it will, but see the 5G towers and everyone's expecting to start marching like a little robot. They're gonna turn you on someday, activate you.
I don't think that's
Okay, we're gonna talk about pigments. This is B9. We weren't necessarily thinking we were gonna talk about it. So one of the things we have discussed before are that pigments have conjugated systems. Those are alternating double and single bonds. The extent of delocalization of those pi electrons gives rise to the intense absorption in the visible spectrum. You are familiar with many a colorful thing. You know hemoglobin. You know what myoglobin is. You know what chlorophyll is. You know what cytochrome C is. Those are all super colored things. I don't know what cytochrome C is. It's in the electron transport chain. But you want to know cyto, cell, chrome, color. I'm guessing it gives cells some color. Myoglobins in muscles. I don't know how myoglobin and hemoglobin are different. I don't believe they're too different, but I do believe that myoglobin is why meat turns brown and it like oxidizes. If you've ever like had hamburger and then flipped it over, so live that life. We're gonna pay some special attention to hemoglobin at least. Not too scared about that. We'll talk about anthocyanins. Anthocyanins are the dyes that are in berries. We'll talk about carotenes. That's the same one that's in beta carotene, one of the vitamins we saw. We'll talk about how when you look at something, there's a cis-trans rotation. Opsin. I don't know. We'll talk about it. Hopefully we'll know what we're talking about. And then some things that maybe some of you have seen before that will make this unit interesting are how oxygen binds to hemoglobin. So some of you may be aware that hemoglobin has quaternary structure. That is to say that there are 4 proteins that are together, 2 alphas and 2 betas. Each of those proteins that is folded together has one heme group in it. Each heme group can carry 1 oxygen. The way hemoglobin works is it becomes stickier to oxygen as it collects oxygen. So an empty hemoglobin is the worst at attracting oxygen, and a nearly full hemoglobin is the best at attracting oxygen. That doesn't seem perhaps as you would expect it, but many of you have seen this story before, so we'll have, someone like, go ahead and tell it. But we're also gonna talk about the distinction between fetal hemoglobin and normal hemoglobin and how that might work to facilitate oxygen transfer in the placenta. I hope it's super fun. We'll see. So again, the main classes that we'll talk about are keratines, porphyrins, and anthocyanins. Porphyrin is not a word that you guys are familiar with so much, but hemoglobin and chlorophyll are both examples of porphyrins. Porphyrins are nitrogen-containing macrocycles associated with color. We call the part that absorbs light a chromophore. I used that when we were talking about Beer's Law last week. We talk about the alternating double and single bonds as conjugation. You can also talk about it in terms of pi electron delocalization. I don't think that Aditi has to spit the word conjugation, but she might have to read it. If she wanted to understand it again, she's looking for alternating double and single bonds that mean that the pi electrons can move about in the molecule. Something that you learned a long time ago is that you absorb the complementary color. So if beta carotene is orange, what does it absorb? Blue. You hate colors? Any questions about that? Thoughts? Where's the color wheel if we have a moment of stupidity? It's in the data booklet. And that's spelled wrong too. Terrible. See that? Red? Is that a trick question? Okay. What's the metal in hemoglobin? What's the metal in chlorophyll? It's like she's looking at something that has the answer. I don't know what it is. So chlorophyll is green and hemoglobin is red, but they both have the same organic structure around them. So what's affecting the color? The metal. It's traditionally a transition metal, but it's hard to call magnesium a transition metal, so we shall not. What does myo mean? It does mean muscle. Oh, it's actin and myosin, right? Okay, so we met carotenes before. Again, we met them in the context of vitamin A. You see that alternating double and single bond section of conjugation. Take that oxygen out because it doesn't belong. If it appears yellow, what color does it absorb? Purple. So in addition to having chlorophyll, plants have quite a lot of carotenes. Did you guys do a chromatography experiment with leaves in bio honors or IB?
Yeah.
Have you ever done it? You didn't do it?
To put in like the solution and like see how much the treatment's traveled out. It's like much.
Like we did it with different types of.
So yes but no. So you're watching capillary action with the celery going up. So you're taking usually a dyed thing and letting the celery suck it up. But what I'm gonna do is I am going to rub a leaf, honestly, on a piece of paper. I'm just gonna rub the crap out of it. And I'm gonna try to get the dye that's in the leaf to stay in my paper. And then I'm gonna put my paper into, no, it should have a lid on it. Y'all can draw your own lids. Onto a piece of paper, here's my stuff, and that same thing that was sucking the water up the celery is gonna suck the solvent up this piece of paper. And when you look at it again, traditionally you should see 3 bands as well as the solvent front. That's when I'm gonna take it out, I draw it with a pencil. The 3 bands are traditionally carotenes, chlorophyll A and chlorophyll B, but it does depend on what age your leaf is. When we're really good, we do it in the fall, so we get some nice red colors in there. It's kind of cool to see some different shades. How would I calculate an RF factor, a retention factor?
Where the line is and where it starts. Or where it ends, whatever.
Perfect. So the retention factor, it's in that spirit of ATM machine and bit number. I apologize, we say RF factors sometimes. It's just the ratio of how far one of the spots moved, so this distance, divided by the total distance that the solvent was able to move. we can compare RF factors between different experiments and identify that in this solvent, chlorophyll A has an RF factor of about 0.45, and so all of us have a leaf that contains chlorophyll A. So it is a way of qualitatively deciding that we have the same material. I believe that we spoke about how if you buy grass-fed meat products, the fat is typically yellow, and that's because of the retinol in the fat.
What about fat that's like kind of whitish?
So that's an animal that probably was on a grain-based diet, didn't have this vitamin, and so you don't see the pigment in the fat.
Is it better to have the pigment yellow?
I mean, I assume the vitamin A is better for the animal and for you, but I don't. Just gonna look at vegetarian in the room. We're gonna be like, yes, yummy, yellower. So this is retinol. It absorbs purple light. It appears yellow. This is its buddy, beta carotene. Why would it absorb a different color of light? I'll go back. If you want to count, there's 1, 2, 3, 4, 5, 6, 7, 8, and then alcohol, carbons. I'm going to look here. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, and then the other side. So why do you think these might absorb different colors? This is a longer chain, so because it's a longer chain, the extent of delocalization is greater. And so when I'm holding my recorder down and all of the little holes are plugged up, do I make a lower note or a higher note? All the holes are closed, or it's an open guitar string, it's an open note.
And it's lower.
It's a lower note.
Lower.
So would this absorb lower energy or higher energy? The answer is on there, so we just gotta figure out why.
So is this like, what was the note analogy about?
So, to me, when we had just the carotene that had 8 carbon chain, versus here we have, I forgot what I counted, a 16 carbon chain. So to me, this is like a recorder that's half held down, or a guitar note where I'm gonna hold the fret halfway, and this is one where it's completely closed.
So the open is like lower, but the 8 one is like lower. Did you notice or higher? Because
Higher.
it's a higher note because you're like on a higher fret.
Yeah.
But then the more double bonds, so it has lower energy.
Yes.
So it goes into like bluish color, so it's more.
Yeah. Yes. So the extent of delocalization is larger. Maybe I shouldn't try to do this in music, maybe I'm out of my depth. If the extent of delocalization is larger, the pi to pi transition will be easier, or the energy transition will occur at a lower energy. And so you can see that this one, this larger molecule, absorbs lower energy blue light as opposed to violet light. So as we make the extent of delocalization greater, the energy goes down.
Just because there's more number of boxes.
Yes. Box is bigger is what you'll say, in a year or so.
What's the difference between retinol and ethanol?
Sure, we're gonna have to learn about it when we talk about rhodopsin and seeing things. I'm not ready.
So it's different.
They're different.
Okay, so retinol is not on it.
Cis-retinal. I think that's what you call it when it's in like what we're gonna see. So I think that you have the vitamin form, not the form that your eyes use directly.
So what's on the frequency?
They call that vitamin A. Oh, sorry. They called this vitamin A. Are they not consistent with the data booklet? Like, it's not the first mistake. But this is trans.
So all trans retinol.
I have no answer. So I know we're gonna talk about that cis-trans isomerization.
Oh, it's here. There. Oh, okay. So retinol is different. Oh, the only difference is just the configuration.
That's my
Oh, no, it's completely different.
completely different. How exciting for us all.
Well, it's like some,
Oh, two are an aldehyde,
yeah.
and this one, yes. So one is oxidized form of the other, at the very least. I'm sure we'll get there. I can't wait. That's the nitrogen-containing macrocycle that you find in hemoglobin and in chlorophyll. The metal just hangs out here in the center, making some cute coordinate covalent bonds in a square planar kind of arrangement. What else does iron carry? Yeah, I feel good about that question. What else does iron carry?
What's the first thing?
The porphyrin. Oxygen.
I thought that was the worst.
And so you can see that there's a spot that an oxygen can bind right here. Oxygen has a lone pair of electrons. It just makes another coordinate covalent bond. What do you think about the relative strength of a oxygen coming in and a carbon monoxide coming in?
Carbon monoxide's higher.
Carbon monoxide does bind better. How do we know that? Because people die. I mean, that's not a great way to know it, but like it's an empirical sort of, if you have carbon monoxide poisoning, it's because the affinity for your hemoglobin toward that carbon monoxide is so much higher that it can't let go. And so you suffocate. You are no longer able to bind oxygen and move it around your body. Now you notice the left picture and the right picture are two different resonance structures. And so resonance structures always lead us to believe what is present. Electron delocalization. This time the delocalization is in 2 dimensions. So instead of being along the retinol, thanks for that, it's going to be in the box. That is 2-dimensional. And so this compound by itself is a very, very purple color. Porphyra means purple in Greek.
What does the arrow that's going both ways mean?
The arrow between the 2 ways means that those are 2 resonance structures.
So any one of those is acceptable?
Yes, and no one will ever ask you to draw it. It's surprisingly hard to draw. We use the word aromatic to mean alternating double and single bonds in a ring. The opposite of aromatic is aliphatic. All of the organic compounds that you are familiar with are aliphatic, starting with a D. Aliphatic. A famous example of conjugated molecule is naphthalene. Is anyone old enough to know what naphthalene is? Naphthalene. I don't know, like where would I find naphthalene in your house? But you know, I'd probably find it in your grandmother's house.
Like a baby powder? Is it what makes them smell like grandma's?
So I believe it's what we use in mothballs. So if you've ever put mothballs around your house, I think that we usually use traps these days, but they do have a scent ingrained in them. This molecule, it's known for having a smell, and so I think that's probably where we got the word aromatic. But let's say it's at best silly. What are mothballs for? I don't know.
Are they actual
Balls?
moth?
I think that they repel. Like I think the moths simply don't like this. So like, peace out, I'm going somewhere else. Whereas now we use traps where the moths are like, ooh, smells delicious, and then they're stinky, and so the moths
I
stink.
don't know, mothballs, and like, I don't know, like, so, are they made by moths or are they put so that moths?
No, they are
don't
put
go?
so that moths do not mess with your clothing. So you used to put them in your closet. I was gonna say pantry. I don't keep clothes in my pantry.
My grandma loves mothballs.
Thank you.
They sound,
They smell bad though.
I feel like they could have picked a better name, 'cause it's like mothballs, it sounds kinda off-putting. Like, it sounds like they made like a nest and just like left it. I don't know how moths.
Do any of you use cedar in your like drawers or closet? It's the same idea as cedar, just a deterrent.
My grandma
Well, now
smells
they're on
mothballs.
supplementation, you're supposed to go, like, solid.
Yeah.
Mothballs does not smell like that.
Toxic to moths and moth bite. But they stink, and so I think we call it aromatic because mothballs stink.
My grandma just smells like mothballs.
No.
I can tell it when I give this to her
It's like
because it
chlorine.
smells like mothballs.
Well, look, so there is a heme group. A heme group might be found in hemoglobin or myoglobin. There are coordinate covalent bonds between the nitrogens of the porphyrin, forming this square plane. So this right here is the square plane of the heme group. I said, what else might the iron hold? We eventually got to oxygen. But it's also stuck in that protein. There's a histidine that forms the 6th coordinate covalent bond. So what is the shape around iron in oxygenated heme?
If it has 6 coordinate covalent bonds, like 1, 2, 3.
Yeah, 6. Octahedral.
Oh, like that's for sure.
So again, you have this in the plane and then an oxygen up and a histidine down. Again, we're praying for questions we can answer. Emma and I, we don't study the biology, but I can tell you that's octahedral and not know anything about how it works. Yummy.
Do people eat it like that?
I eat it like rare. Yeah.
Where's that?
I like it still with skin.
When you change the oxidation state of iron, why do you change the color? We are not doing a Maillard reaction here.
That's what it is.
Okay, that's what they're showing in the picture, but that's not what they should be showing. They should just be showing aged meat. Again, my question is, why does oxidation state or charge or oxidation number change the color?
It's like rust, right? But the same idea. They just like turn it around.
I would not describe rust as a coordination complex. I'm not giving it to you. Like, I want an answer straight from topic 13. There are 3 things that allow me to change the color of a transition metal complex. They are number of ligands, the shape, and the oxidation number. So I was curious why oxidation number would matter. Why does it matter if it's iron 3 or iron 2?
That's how many less or more electrons it has. So if it has like, in terms of like bonding, I think like if something has like more or less electrons, it's more available to bond with other stuff, which can.
But this is a Lewis acid, so does it want more electrons or fewer electrons to be its best electron pair acceptor?
It's a plus, so it won't want less or more.
So this guy wants more,
Because it's a
like it has a stronger affinity for the ligand.
Yeah.
If it has a stronger affinity for the ligand, is that gonna make the crystal field splitting energy higher or lower? That's right, because the bond is gonna be stronger. So Quinn is convincing me that what I'm doing in moving the iron 2 to iron 3 is I'm changing the crystal field splitting energy to show that the affinity for that ligand is higher because the charge is more positive. Again, praying for questions I can answer. Red blood cells don't contain DNA. Same and like why? Why do we have cells that don't have nuclei?
Just
here's a hemoglobin. We talked about how it had 4 units. Remember, those 4 units refer to something known as quaternary structure. That is the interaction of already folded groups. You can't tell that there are 2 pairs that are the same, but you can see that they are labeled as 2 betas and 2 alphas. Nestled into the heart of each one of these, near a histidine residue, presumably, is a heme. We refer to each one of those as subunits. There are 4 subunits, and just because biologists need lots of words for everything, that little hemoglobin molecule or heme molecule in the middle is known as a prosthetic group. I would not get that mark. Yeah, Samarachi.
2 questions. First, could you go back to the first hemoglobin slide?
Yes, ma'am.
And then second, if red blood cells don't have any genetic material, how are they like made? 'Cause I know your bones do.
That's exactly what I'm saying. Like, where do they come from? How do they?
Differentiate those.
If they're just protein, there's no other.
But even proteins need genetic, they're made through what's it called? Central dogma, and that requires genetic material, no? Like protein synthesis.
That's in the
They
bone.
Sorry?
Isn't that in the bone marrow?
You know what?
Bone marrow?
Yeah, it's made in the bone, but my thing is,
How? Wow.
I don't know, I think it's like the first phase in order to like.
Really? I legitimately like.
I think it was like baby red blood cells, they start off like small amount of DNA, but then when they're mature, they lose the DNA or whatever, like genetic material is needed or something. I don't know, it's weird, it's odd.
You wish we knew things like this?
Basically, stem cell flies into many different red blood cells and then releases all of them. It's a full spectrum.
I understood all those words. I just don't know.
They start off in the nucleus, and then the theory is that once they get mature enough in order to like be and be your red blood cells, and be considered red blood cells, they like nuke the nucleus. That's essentially what it says, and then it's enabling the red blood cell to contain or carry more oxygen, or more oxygen carrying hemoglobin.
Red blood cells like us exist to die.
if they lose the DNA and they become adult form, then how are more made? Then they're just all.
Because they don't make each other, right?
Okay, so it's just all being made
They
by
don't
itself.
like how like cells with the nucleus.
Yeah, I know what you mean.
Split. They don't do that. They just die.
No, their cells don't spit that out or something. So I think that must just be.
It's one cell
Bone
kills
marrow.
many red blood cells and it splits up.
So here's another picture of hemoglobin. You can see it without the oxygen, so the porphyrin is actually buckled. So here's the histidine residue coming up. The 4 nitrogens are not quite square planar. And when the oxygen comes in, it makes a true octahedron. In the process of doing that, what we did was we moved the histidine residue. In moving that histidine residue, we have made somehow this structure have a higher affinity for the next oxygen. So it's like a mechanical thing that occurs. We refer to this effect as cooperative binding. The first oxygen changes the structure, and so makes it easier for the next oxygen to bind, which in turn makes it easier for the next, and finally for the last. So we can draw a picture with a sigmoidal shape. I don't know the difference between logistic and sigmoidal. So on the y-axis, I am showing you the percent saturation, which I assume is how many hemoglobins have pressure of oxygen. Does that make sense to anyone? Yeah, it doesn't make sense to me either. So, like, what I like about this curve, and then what I don't understand about it in different parts, is that when the pressure of oxygen is low, the rate of me saturating the hemoglobin is low. And as the pressure of oxygen gets higher, you know, the rate gets higher. But at some point, some of them are full, and so it slows down again. So I can't draw cardiovascular respiration. Like, imagine the heart, you know, you like cycle twice. Imagine that I know what that means, I almost know. So you are deoxygenated hemoglobin. So you're coming back from the body, right? You're going to the heart. What do you want to do?
Like, you're deoxygenated, so you're gonna go. Are you asking like the actual process of
Okay, help me out.
No, I don't know what you're asking.
I'm saying as a hemoglobin, if my job is to distribute oxygen, like how would it be efficient?
In the capillaries of the lungs, it goes as like each red blood cell passes through like the single layer of thickness. That's where oxygen can come in. The hemoglobin onto the red blood cells.
Okay, so you're describing how in the lungs we can reoxygenate this, and I guess what we're gonna find out is that we are gonna have an easier time oxygenating things that are already oxygenated. So we're frequently gonna make full ones, right? And it's gonna be easier to fill them up because they have a higher and higher affinity for it. So something that only has 3 is gonna have a relatively easier time getting to 4. So now it's gonna go through your body. And when hemoglobin is full, again, it has a high affinity for oxygen. So is it gonna have an easy time giving up that oxygen? It's not. Because like if I had a full wheelbarrow and I was really struggling with it and I found a place to drop some stuff, I would drop it at the first place that was available. And so I feel like whatever is straight out of the heart would get all my oxygen. I'd be like, good luck rest of the body, I'll be back next time. But instead what happens is that when you get to that first thing that wants oxygen, you're still at your highest affinity for oxygen. You're like, I'm not gonna give it to you, I'm gonna keep going. It's gonna take me a while to give up that oxygen, so there is a chance that we could distribute it around the body. What? It's really good.
And I think that's why your important organs are like in the central area of your body, not like your heart's like here, because it's more efficient.
Okay, let's see. There's just not enough words. Lord. I'm gonna overlay what happens for a baby. HB or HG? What is it? So that curve appears to be higher. So at a given partial pressure of oxygen, what's true about the amount of fetal hemoglobin that would have oxygen? It's greater. So if we were to have mama's blood and baby's blood on opposite sides of a placenta, what would happen to the oxygen? It would move to the fetus because the fetus has a stronger affinity for oxygen than the mama. And so that is how oxygen gets to your baby, because while, you know, baby might be in the center of your body, it's a ways away. And so we needed to have a way to make sure that baby got the oxygen. Don't know how. Let's hope this is just some chart reading. Blue line is you at 37 degrees. Green line is you with a fever, a really big fever. Don't get a 40-degree fever. That's a lot of fever. What is that going to do? hemoglobin is being less efficient at this high temperature? Is there any reason that that could help me?
Help if having a lower.
Because sometimes my body raises its temperature. I don't think it does it foolishly.
Because when it has to kill off your fever, you get a fever when you're sick, so that your internal body temperature is higher than the like ideal temperature of whatever is like infecting you, so that it goes down.
Like when you're, oh, okay, that's maybe a better example. So you're saying that I'm gonna kill the greenies at a higher temperature.
Like the bad stuff that babies.
So all of my biology class was told in the context of greenies, which were made-up organisms that had, genetics similar to real things, but they lived in Schlitz beer, and so like the whole biology class was about greenies. Good. Okay, so you suggested this could have something to do with exercise. So when I exercise and I start to get hot, does it make sense that I would want to have a lower ability to hold on to the oxygen so I can give oxygen to the muscles that so wanted it? I think that's compelling. How about pH?
Actually, CO2 is acidic, so
So
more pH, more CO2 makes it more acidic.
when the pH is lower, or when the concentration of CO2 is higher, what am I about to do?
When your CO2 is higher.
I hope I'm not gonna die.
Oh, right.
I really hope it's not death. Like, so like, I feel like, where's the vena cava?
Oh, actually.
It's on the bottom and top of the right side of the
Is that like one of the last places my blood will be before it gets reoxygenated?
first. It goes into the
That's
heart.
how it
through the-
enters.
Harder.
Right, so it's
Harder.
empty.
I mean, if
It's where it brings the blood in.
aorta is the last
There's
place
one
that the blood
superior
is.
one and there's one inferior. So the one on the top brings in blood from the upper half of your body and the one on the bottom brings stuff in from the lower.
Okay, I'm gonna draw you a simpler picture of my understanding. We're gonna have heart and lungs. Okay, on this side, we're gonna have full of oxygen. And over here, we're gonna have empty.
Yeah, like,
I don't get you. Telling me it's not exciting, right? I'm telling you that's your biggest problem.
no, it's correct. That's
Yeah.
in the body.
Pretend you're standing in the hallway.
Some physics.
Okay, I'm gonna try again. So I am cellularly respiring, right? So my body is hungry for this oxygen. When my body takes in the oxygen, what is something that it might leave behind? Carbon dioxide. So here, I would say I'm empty of CO2. And here I would say I'm full of CO2. Because when I get to the lungs, one thing that I've got to do is exhale CO2. Another thing I've got to do is, inhale O2. Again, this is a simplified picture of some things I don't understand very well. My question was, when I'm over here, when I am full of CO2, when it's about to make it back to the heart, what do I want my hemoglobin to do?
To take out some more.
Like really, the oxygen,
In the full of CO2 place.
So when I'm in the full of CO2 place, I would believe that I would want my hemoglobin to start letting go more readily.
So that you're oxygenating the
So
body.
I'm oxygenating my body and not bringing back full hemoglobin to my lungs. So I was trying to argue, and y'all can correct me clearly, that that's perhaps why this curve has that shape. When you are full of carbon dioxide and presumably just about out of oxygen, you would perhaps like it to be easier for the hemoglobin to give up what remains. So when I see full of CO2 and empty of CO2, I see about to exhale. Let's all go show that to Miss Meyer. Let's show it to Miss Lee so she can judge me. Let's see what this fine sentence says. So it says slightly decreased pH and increased partial pressure of CO2 act as non-competitive inhibitors, allowing more oxygen release in venous blood where tissues may be oxygen deprived. I like that. So the veins go toward the heart. That's why I chose the vena cava. It sounded like a vein to me. And so we have deoxygenated tissues that are hungry for it. Yes. Because those are related. Because the reason that the pH is changing is because if you put CO2 in water, it makes carbonic acid, which dissociates to the part I understand of this story. I don't think I'm gonna pass the physiology part of your IB exam, I'll be honest. And have you done the strings, the yarn, the red yarn and the blue yarn and traveling through the heart? Yeah, I've heard them doing it in the hallways. Like, I really should be listening because it's clearly something I don't understand. Ooh, this looks like an interesting picture I do understand. Let's go. We talked about the relative affinity for carbon monoxide and oxygen. How do you feel? Good, you should. You should have a carbon monoxide detector in your house, in your bedroom. Why is this a competitive inhibitor?
It stops the oxygen from binding.
Takes that same binding site. How do we decarbon monoxide you? What would you do?
Like, is it something that's done?
My first is to activate a charcoal or something.
No, they put you in a really oxygen-rich environment. Like, yes, it is way more, it has a much higher affinity, but it is possible, like, it's still reversible. So every time one leaves, you just play a statistics game that there's a whole lot more oxygen present than carbon monoxide. Very bad for you. And there's another typo in these goddamn slides.
It's the same way they treat nitrogen.
Yes. Yes, I thought we might get there. Let's hope we don't. You don't know anything about cytochrome C, right? Cytochrome C oxidase.
I've heard of it like in a bio.
Okay. I don't know, I just have a slide of it, I don't know what I'm supposed to say about it, so let's see what we're supposed to say. So cytochrome, so I have an iron in the center of a porphyrin, there's a histidine holding it in place, it's got an oxygen on it, and now it's got this other jazz coming up. It has a copper. Those are two more histidines, and that's a tyrosine. Definitely in the electron transport chain. I don't know, what would we say about it? Does it look colored to you? Yes. Good, it looks colored to me. What's the shape around this copper? Sure looks tetrahedral. It's got coordinate covalent bonds to 3 nitrogens and an oxygen. I clearly don't know a question about it. Hey, hemoglobin and chlorophyll are like neighbors of each other. So you can see some changes are going to be that this hydrogen becomes a carbon-20 tail, that this carboxylic acid becomes an ester. But on the whole, the molecules are really, really similar. You are not responsible for knowing any part of that molecule aside from there's an iron in the center, there's a magnesium in the center, and it's in the data booklet. Can't imagine what they would do. addition to chlorophyll, there are other accessory pigments that include the carotenes. Why do plants absorb light? To make food, they need it for photosynthesis. They'll take it all. So in addition to the chlorophyll, we have accessory pigments. Those are typically referred to as light harvesting antennae. I was interested in those in grad school. I'd like to know more about that. So when you guys are rich and famous, please come back and teach me about that. Very interesting to me. What do you know about an electron transport chain? That's what I know about it too. What's P450? Oh, I like those words. P450. I feel like it's a thing. Not in this thing. So in the chloroplast, we have the thylakoids that line up into grana and the stroma.
This is what I know.
What on earth do we have here? That's a thylakoid, and here's the membrane of the thylakoid, and this is where electron transport is going to happen. What do you think's in photosystem 2? What? Chlorophyll, oh good. Pigment. And what do you think's in plasticlinone? What do you think's in cytochrome? What do you think's in plastocyanin? It sounds like anthocyanin to me. Cyanin sounds like cyanide. Cyanobacteria. What do you think's in photosystem 1? What do you think about ferredoxin? I think what we have to get over is that we don't need to know the details of this, thank God. But there are a lot of different things here that absorb the light energy. What does that look like? So I'm a chlorophyll molecule. I absorb this color of light. That means that I have an electron up here at that energy level. Where is the electron going to go? What else is going to happen? What does that look like? So is it a redox reaction? Yeah. Yeah, it's a redox reaction. So this electron will be given to something. So this molecule would be oxidized, and another molecule might be reduced. And then the high energy electron there could find someplace lower to go, and then we like promote it up again. So like the energy was going down, you know, we pop it up. I don't know. We'll have some practice questions next time. I can't wait. Hope I get to draw this picture again. Oh look, oxidation reactions, like we know what we're talking about. Move on, pretend we talked about that. Oh, anthocyanins are back in business, baby. Anthocyanins are the pigments in fruits. Among my father's witty sayings, blackberries are red when they're green. See, when blackberries are unripe or green, they appear red, and as they ripen, they turn black. It's because of the anthocyanins that are present. What do you notice about anthocyanins? They have alternating double and single bonds, electron delocalization, and consequently have beautiful colors. We think that we like these colors because birds like pretty colors. To me, that's what spreads fruit, but you can, again, biology me up if you would like. But we can also use them as delicious pH indicators. It's so good. So if you ever did red cabbage juice, you would know that red cabbage is actually just another anthocyanin dye. And what you're watching is the anthocyanin dye undergo repeated protonations or deprotonations to change its color. So in the fully protonated state, we refer to the anthocyanin dye as the flavillum cation. Where, oh where would I find that? The answer is the data booklet. The answer is, I mean, how the could you possibly know? It's written right here. So the word flavillum cation is there because no one expects you to know that. The structure of the flavillum cation is there. No one expects you to know that. And the fact that it's red is there. So this information is straight from your data booklet. Which one of these absorbs lower energy light? Okay, so he absorbs orange, which is lower energy light. And he absorbs green, which in this case is higher energy light. And what I notice is that that one's got one little bond extra in this alternating pattern. The extent of delocalization is just a tiny bit bigger, and with that extent of delocalization being just a tiny bit bigger, we get a change in color and a lowering of energy. Isn't that lovely? If we use this as a pH indicator, what color would it be at the equivalence point of the world's best titration? Purple. When we use indicators, which are all weak acids, we talk about how we can set up the weak acid equilibrium So when this system is quite acidic, what color is it? So if we increase the concentration of acid, Le Chatelier says the stress that we have exerted is too much product. So the release of that stress is to shift back and turn red. On the other hand, if we add base to this system, the base is going to react with the hydronium, making water. That's going to take this away. Which way is the reaction going to shift? Forward, and it's going to look blue. But at the equivalence point of a titration, Karthika told me that I was looking for a mixture of red and blue, and that mixture would honestly be so that the concentration of the indicator was equal to the concentration of the deprotonated indicator. And so that means that the pH would be equal to what and why?
7.
Not 7, but the pKa. And that's because of Henderson-Hasselbalch. Remember, Henderson-Hasselbalch told me that while I was in option B, I should use my equation, as often as it possibly can, because it's a freebie. If these two values are equal to each other, then the log of 1 is 0 and the pH is equal to the pKa. We're at the half equivalence point. So we're imagining a titration, and what we're trying to do is set the equivalence point of our titration to be approximately equal to the pKa of our indicator. So why? So as fruit ripens, the acid turns into a sugar. And so we're going to notice the color of the red turn more and more purpley blue. Oh my God. You have 9 minutes to cry. We're going to talk about stereochemistry. I don't know. My plan for next class was to give a test. My plan for this class was to review for the test. Suddenly I'm talking about myoglobin. I'm drawing a picture of a heart and lung because I don't know how to make it work.
I think it's a bit better.
Like they make a machine, right? The heart and lung machine. It has to have one in and one out, right?